They're continuing now with the 2019 Module 2 for BioUnit 2, right? 2A. Impaired glucose tolerance is a risk factor for certain diseases. An oral glucose tolerance test was administered to persons with normal glucose tolerance (NGT) and impaired glucose tolerance (IGT-1 and IGT-2). Blood plasma glucose levels were monitored at 30-minute intervals for 2 hours. Alright? So we end up with this table with the important information in here. Alright? On the grid on page 11, plot a line graph of the data provided in table 1 with time on the x-axis. So if that is the case, how are we going to provide this graph? So I said that time must be on the x-axis, not true? Alright? So if we were to draw a graph like, oh my gosh, don't watch the thing. Alright? Don't watch it. Alright? So if we're saying that time is on this axis now, right? So time is on this axis, right? If we're gonna have NGT, IGT1, and IGT2, that means that we're gonna have different things to represent the points, not true? So remember to do that, so have to distinguish between them, right? So since they're line graphs, you could use different lines, right? Or you could use different points to represent the differences. So NGT could be your dot and circle. Your IGT1 could be your cross. Your IGT2 could be, let's say, a cross, but with dotted lines or something like that. You need to show the difference between them. All right, does that make sense? Do you understand what I'm trying to say? So if you're going to do that, you must provide a scale, all right? And your scale will you must provide a scale rather. That's fine. But you also need to have the key rather. That's what I was trying to say. The key with showing what represents what. So what represents NGT, what represents IGT, what represents IGT, well, 2. Alright? That's what we're looking at. Are we all okay? Not hearing anyone. Everything is fine, guys? Okay, lovely. State 2 key observations that can be made based on the results of the study. What 2 observations can you see here? So we're seeing NGT. I don't know if I'm straining anyone's eyes, so let's zoom in a little bit more. Alright. So we have this NGT, IGT1, and IGT2. What is the difference here? So one thing I can note here is that the peaks in IGT1 and IGT2 are higher than that of NGT. Right, so that's one thing I could state here. So one thing I see here is that we have higher glucose levels in IGT1 and IGT2 compared to NGT. Right? That's an observation that I can see. Now, what I normally do, right, because it said based on results, we're not going to just state our observation. I would open a bracket and show exactly what I'm talking about. So where did I see that observation? Based on what results? We must show it that it's based on those results, right? So we're seeing that the higher concentration, so we reached what, 10.7 for IGT1, and we reach what, 12.6 for IGT2, right? Compared yet again to 7.8 in NGT. That is how we do it based off of the results. So we mustn't just state what we're talking about. We must do it based on the results. Alright? Another thing, what could we look at here? Mm, mm, mm. So we could look at the graphs here. With this graph, we're gonna have 5 to 8 to 7. No, it should be 8.8 rather. Why did I put 7.8 there? One moment, let me fix this. If we look at these results now, we reach a peak around 30 minutes here for NGT, right? And then it goes back to 5.2. It goes back to the baseline. That is what I would call it, the baseline there, right? While IGT now reaches peaks, right? Higher, even though it's higher peaks, it takes a longer time to reach peak than the NGT, and that's the same for the IGT. So I would say no, that it takes a longer time for them to reach there, or what we could say it's a delayed return to the baseline. So it takes a longer time to reach the peaks for IGT one and two, and also we could say we have a delayed time or delayed return to the baseline values, meaning where we start from. Baseline means where we start from, right? For that value. Right, and obviously you just look at the page and get the um make it based on the results there. So tell us what you're seeing, and then now tell us what is happening in the table to explain what you see or why you say what you see. Alright? Alright. Are we okay with everything so far? Are we okay? Are we great? We're lovely. Are we good? Let me check. The persons who are talking stop talking. That means that this meeting becomes silent. Okay. Alrighty. So I have that there. I will discuss slightly about the graph. Anushka, answer this question from the please since you like to laugh. Explain the mechanisms by which glucagon and insulin function to maintain a normal concentration of glucose in the blood. Go ahead and Anushka, give us a go at this, right? Before Mr. Bovell goes. So divine and go ahead, Anushka. Wait, wait, don't. You're sitting here laughing, you know. Okay, go ahead, Bovell, and then Anushka to plug any holes in the response. Go ahead. Hold on. Hold on one moment. Before you start becoming an encyclopedia like we normally do, right? Hold on. Right? Let's structure it. We explain the mechanisms, right? There are 5 points in the mechanism. What are the 5 points? So the 5 points we're looking at. Try to structure the question, and then we put everything neatly so we don't miss anything at all. Organizational structure. This is why we come studies. First one. What is the first thing? hmm So we're looking at the stimulus detection, stimulus slash detection. So what stimulus are we looking for? Okay, so we're not putting detection there because my brain moves it. Alright, so we're looking for a rise in blood glucose levels. We're looking at which glucagon, what does glucagon do? How does insulin do? Insulin and then glucagon. Because glucagon come first in the question. Sorry, I'm that's why I am. So because it come first in the question, go ahead. Okay. Wait, one. Hold on. I can only type so fast, right? Um, this is detected. What are we looking? We're looking for osmoreceptors, right? Is osmoreceptors? Okay, lovely. I want to know if you know exactly what you're talking about. Go ahead. So repeat what you're saying. The detection one. Mm-hmm. Okay, hold on. Langerhans. It's Langerhans, right? Alright. So that's reception, right? And then what we're going to be looking at now is the regulation. What's happening now? So after the detection, what happens directly after? Okay, glucagon into the bloodstream. Let me just fix up everything. Mm-hmm. Mm-hmm. Hold on. Hold on one moment. Right. So we're taking the anacyclates already. Okay, go ahead. I'm trying to think about what you're saying while I go through it. Go ahead. All right. Okay. Any issues there, Divine? Okay. Okay. Mm. Alpha cells. Okay. No problem. It's fine. So caught that before going to the exam and put it, right? So alpha cells that. So oh my gosh. Alpha cells will be that. Right? So everything else here is fine. Let's look through everything. Is that fine? All of it is fine. Let me just take this just for safekeeping and drop it in the meet chat. Alright, so you guys can have that. Alright? Let's drop it here. Oh, why would I? Okay, I'll send the documents. um, oh, my apologies. I need to do that. But you need to. The thing is, no. In order for me to send the document, you need to fight me. All right. Yes, exactly. All right. So let's change this up now. So we do for glucagon, right? So for insulin now, what are the differences now? The insulin would now be high blood glucose, right? Okay, so we should put that hyperglycemic. Yes, hypoflow. Oh my gosh. So spelling is something that I don't participate in, obviously. You know when oh I heard actually. Okay. Okay, so I have that there. So we're having high blood glucose concentration there. Go ahead, Jonathan. I think Jonathan raised hand or was it Bovel? Someone's hand is still risen. Oh, no problem. All right, so go ahead, continue. Mm-hmm. Mm-hmm. Hold on. Hold on one moment. I'm trying to fix this thing. I think. Can you guys see what's occurring on the screen? Because it's because I'm wondering. I want to understand what this thing is doing. Let me just put it over here. You know. Okay. But you see, as a male, I'm very stuck in my ways and very stubborn. So since sex box is actually already made, I'm gonna have to continue. All right. So, but okay. So I have that. I just want to like make this simple differences, right? So there's beta cells that secrete something, all right? Beta cells secrete insulin, right? As you were saying, Bovel. All right. So what is the reaction now? So what's regulation now? Where are the insulin? Where are the insulin receptors? No, what organ? That's what I'm asking. The liver. Are we sure? Okay, but we can just go ahead. Go ahead, Christine. Liver and the muscle cells. Okay, so we're having multiple responses. All right. So what is the activation? So after it binds to insulin receptors, right? What happens after that? What do we have happening? Mm-hmm. 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 Okay. Mm-hmm. He's just adding. He's just adding. He's just being himself. I you guys have said that. Lowering blood glucose concentration. I'm just gonna add that at the end of each thing, right? So lowering the concentration there, and then in this over here now it would be increasing. I'm not going to touch this text box again because I don't want it to disappear. All right, so we understand that. Comment on the clinical significance of the presence of glucose in urine. Okay, interesting. Ooh, which one? It doesn't have to be diabetes specifically, no. Go ahead. Was it okay? So what's the difference between type 1 and type 2? Hmm. All right. So okay. So I'm gonna try to say this once, right? As a cake food and nutrition past student, right? Type 1 diabetes is an autoimmune problem that sees the destruction of pancreatic beta cells. Right? So type 1 is when we have no insulin production from those destroyed beta cells. It usually happens suddenly in childhood or in young adulthood. Repeat? Sure. Well, they're malignant cells, right? But they're malignant pancreatic cells, meaning that they're not cancerous. All right. So we have that there, right? So the risk for these are autoimmune things. These are genetic issues for the type 1 diabetes, right? It always requires some type of insulin therapy, right? And this results in rapid weight loss and thirst and frequent urination and all sorts of stuff like that, right? And it is an absolute immune deficiency, right? For type 2 now, we have insulin resistance, right? So they don't respond, right, to insulin. The cells don't respond to insulin, right? And we're going to have eventual beta cell dysfunction, right? So it's gradual. So type 2 normally starts appearing not in childhood, in adulthood, right? But we're looking at that, all right? Obesity, sedentary lifestyle, genetics, and these different things can cause type 2 diabetes, right? And we know that it's managed through exercise, diet, medication, stuff like that, right? So notice that. So type 2 diabetes is often the milder kind. It often goes unnoticed for years. You often find out when you're in your 30s, thereabout, right? It is preventable, right? And it is, for this is generally just the immune resistance and relative deficiency, all right? So that those are differences between the two. Go ahead. No, not. I'm not complete with this question. I'm not complete with this question as yet. Anybody else? Because there's something else. It doesn't have to be diabetes, no. So if you're not diabetic, you can go ahead. Yeah, it can be through that. Sure. Mm-hmm. Exactly. Exactly. Right. So looking at the cause of glycosuria, yes. So glycosuria is caused from hyperglycemia. So Hyperglycemia. Okay, lovely. Right? Hyperglycemia. Right? Go ahead. It comes from uncontrolled diabetes mellitus. So it's really and truly type 1 or type 2 diabetes. Alright? But look at the renal significant. hmm I mean, that could work. So I just want to explain that a little bit more. It could also be just renal glycosuria, meaning that you have a kidney disorder. Yeah, you can just say that. Alright? Um, you also have, this is gonna be like a way out, right? But you also have alimentary glycosuria, right? Alimentary, we know what what alimentary speaks to, right? Alimentary, the alimentary canal. Repeat. Yes. So the alimentary canal is a digestive canal, the entire part of the digestive system, right? So what we call alimentary glycosuria is that some persons have a rapid glucose or carbohydrate intake, right? Some things like that, right? So that could be what we have there causing the glucosuria, right? Mm-hmm. All right. So I think this is the last question before module 3 and before we take our break. I think so. Let me check. Is it? No, it isn't. All right, but let's complete these two. Alrighty. So question. Well, I think it's still question 2B. Control mechanisms involving receptors occur in plants. These control mechanisms have commercial applications. With reference to the properties of ethylene, analyze its action to bring about desirable changes in fruit during commercial ripening. Go ahead. Gas at STP, lovely. One moment. One moment. Um, you are okay. So what you're saying is correct, right? But I completely and wholly never expected that from a KV Unit 2 student. Did you study everything in depth? Everything. Okay. All right. So could you summarize the biochemical effects of ethylene on fruits? Or give me that, the biochemical effects. There are four major effects. Yes. Mm-hmm. 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 Okay. Okay, so we're gonna note that, right? But after we complete the biochemical processes, what happens from ethylene, then we'll go back to the properties because I guess we got lost along the way there. Alright? But go ahead. We're basically done, right? Okay. Alright, so that's more or less it. Are there any questions or concerns about the biochemical processes, guys? Yellow, red, or other colors, etc. Um, and synthesis of um pigments. So look at the lipocene. Is lipos? Lycopene rather, lycopene and carotenoids. All right. For the flavor, as I said, right, it's going to be stimulating the volatile compounds. Go ahead. Mhm. Okay, good. Yeah, so that is the case. That's why we use the pectinases, right? Stimulation, right? So really and truly, the biochemical processes, right, we should know. So the sugars are broke, the polysaccharides are broken down into monosaccharides, cell wall is softening through pectinases and cellulases, right? The color changes because chlorophyll start to break down, right, and synthesize different compounds. The plant will start to synthesize different compounds like lycopene, right, and carotenoids that changes the color, right, and the aroma and flavor development working on the production of esterification or the ramping up of esterification within the fruit. All right. Another thing about the properties, it's a gas, right? So could you repeat for me what you were saying about the gaseous um nature of ethylene and how that assists? Mm-hmm. Mm-hmm. Okay, lovely. So must tire experiences to everything. So that's actually lovely. So it's autocatalytic. That's what we call it, right? So this is what we call autocatalysis. Go ahead. Okay. So low BP and low MP, right? So it won't condense at low temperatures. All right. So our lovely little ethylene, simple ethene. All right. So as some of us are chemists who understand the entire structure, that should be a difficult part of the question, right? It's just that we need to know the biochemical processes there as well. But chemists do biochemistry as well, so it's fine. All right. So that is the case. That is the case. 6 marks. Right. Moving on now to the last question. While my computer is at 9%. One moment. So let's read the question. Repeat. Oh boy, that's not gonna case. That's not gonna be the case. I'm not gonna put it in now. All right, but while we're doing that now, this question now, the control of the water content of the body is called osmoregulation. Go ahead. Somebody rose their hand. No. Okay. It's called osmoregulation. In the space below, construct an annotated chart to illustrate the mechanism by which the body responds to reduced blood water content at low water potential. Lovely. Your flow chart should identify the effector, receptor, regulator, and response. So I know that you guys are artists rather, and we can construct beautiful diagrams. Go ahead. Um, so the diagram. All right. So let me draw the diagram, and then we can go through all that, and then you can go through and explain each step. All right. Oh, you know it's not going to be beautiful. You know I don't like how you guys do. You know it's going to look nasty, but it's fine. Let's have a look at this. All right. So this diagram is going to look very weird because I am doing it on a computer. So let's see. Um, I'm trying to remember. There was a simple diagram that was in the biology textbook. Oh, should we use that one? I remember how it looks. It's in some notes that I had. So I guess we can use that. Alright, so let's use that. Um Sometimes it amuses me how I can just open my brain and my brain just says, here it is. Alright, so look at this. Hypothalamus, hypothalamus. Right, so the hypothalamus, right, even though that is not necessarily spelled correctly. Um, so we have that there. We can have the pituitary gland, right? We can have the kidneys. Are the kidneys involved in this process? Um, okay, so it sounds like you guys are going way in. Wait, hold on, guys. Hold on. I think you guys are too excited, right? Too excited. Yeah. Okay. It's just because it feels like we're just ready to go with every single detail, and I love that, and I really appreciate it. All right, hold on. Let's look at this. All right. So we don't want to. We're not going into the kidneys. We're not explaining anything like that. Right. We're looking at the entire mechanism throughout the entire body. So even though sometimes in biology it is helpful to look at the in-depth biochemical processes, we must not lose track of the bigger picture. All right. Must not lose track of that. I'll put in the arrows after more urine. Um, okay, so yeah. Or repeat. Okay, so why persons on dialysis urinate a lot? Or diabetics? Oh, why diabetics urinate a lot? Okay. All right. So it has to do with concentration, sugar concentration in the blood. I mean, obviously. Okay, hold on. That's a terrible response. Um. Okay, so with diabetes, would we have a higher concentration of glucose or lower concentration of glucose in the blood? All right, would be a higher concentration. All right. So because of the kidneys, right? So because there's a lot of glucose in the blood, the kidneys will work harder to remove um, all that excess sugar, right? Which means then you'd have the extra urine, right? So the process actually draws a lot of the fluids out of the body, leading to dehydration, and that's why diabetes not only has increased urination but increased thirst, right? So that is the reason. Mhm. That oh, okay, thank you. Wait, was that were you trying to test whether or not I understood what I was doing? Because I do appreciate it. I mean, okay, so well, people are human, Um, so looking at something like this. All right, so let's draw the arrows now. Well, I could just put water. I don't know why I put water. Oh, okay. So, all right, this would go to that. Then we have either this, that, this, it would be this. All right. So I swear to you, this diagram gets better. This diagram gets better, guys. But while I'm doing this, you could have just drawn your own diagram so far. Isn't that very funny? You can just draw a little own diagram on the page while I'm going through this place. Right? Okay, so slavery. H2O. So H2O goes down on this side and H2O goes up on this side. So the amount of water. Alright, so what I'm saying here now is that when a lot of water, right, when the hypothalamus takes a large amount of water in the body, right? So looking at low blood glucose levels, right? It's gonna be detected by the hypothalamus, which will then trigger the pituitary gland, right? To allows us to increase antidiuretic hormone, which is ADH, right? And what does ADH do? Mm-hmm. Okay, so it increases the reuptake of water. All right, so I have that there. All right. So that would now counteract that change. Re- Repeat? No, he didn't state that, but by the collecting done. All right. On the other side, now, when water concentration is high, did you state that it was by the conduct? Wait, you said by the collecting duct. Hold on, the collecting duct. Okay, lovely. All right. So when water, the amount of water is high, right, or a high water potential, right, it's gonna be detected by the hypothalamus, right. Then through the pituitary gland, right, we're going to have the less secretion of ADH, right, and that will cause the kidneys now to decrease its permeability to water, right? Stop aquaporin production, aquaporin action, right? And it allows for more to water to be released as urine. So we just need to draw a diagram and input that information. Go ahead. Yeah, posterior specifically, right? Not anterior. But yeah, so Alright, so what I would do, I would draw the general diagram, and then from here now, I would draw arrows basically explaining the key tips, right? But because it's on a computer, I can't do that well. Right, so for this question, I want you to go and find one of the diagrams online and stuff like that, and look at how that is structured. So you have the general processes occurring on the screen right now, yes, right? But some of them have text box and stuff like that. So yeah, you can draw a diagram, and then from that now you can explain different things on the side, right? So it says to just construct diagram there, and in your chart you need to show the effector receptor regulator, right? So the receptor would be the hypothalamus, right? The effector would be the pituitary gland, not true? The regulator would be the kidneys, and the response, right, is really and truly, right, the decrease of ADH that leads to more urine, or the increase of ADH leading to less urine. So you just need to make sure that's known as well. So you can put in brackets close to the plate, close to the hypothalamus, right, or to different areas, right, to let them know. So this is the effector, this is the receptor, etc. Does that make sense? So you just need to make sure that it's properly done and labeled as well. And that brings us to the end of module